Good afternoon, everybody. Welcome to the Georgia Tech Manufacturing Institute's Fall Lunch and Learn Lecture Series. My name is Billy D. Brown, and I'm a research faculty and director of manufacturing education programs at GTMI. Last week, we had Ph.D. introduce our speaker. She's our Strategic Partners Officer. To start with, I'm going to say a few words about GTMI. GTMI is one of 11 Georgia Tech interdisciplinary research institutes that uniquely focuses on manufacturing research, development, and deployment. We help tackle the grand challenges of today's manufacturers and assist partners in moving innovations from the lab to the market. GTMI has a wide variety of facilities and equipment located on the main campus for basic research and nearby on 14th Street for more applied research at our Advanced Manufacturing Pilot Facility, or AMPF. GTMI's mission includes education and workforce training, collaborative partnerships with industry, academia, and government, as well as thought leadership. GTMI hosts the Lunch and Learn series each semester. This fall, sessions will be held every Monday at noon as live online events. These sessions are excellent opportunities for Georgia Tech faculty, student-level researchers, graduate student-level researchers, as well as undergraduates, and a growing global manufacturing community to learn and share advanced manufacturing knowledge. To ensure a smooth presentation experience, I want to ask all audience members, all of you are already automatically muted. If you have any questions or comments for the speaker, please use the Q&A panel on your screen. I'd like for you to please submit your questions to the Q&A panel as soon as you have them so the speaker can address them throughout the presentation. Today, I'm pleased to introduce Dr. Jay Hoying, who will discuss robotic fabrication of vascularized tissues. Dr. Jay Hoying is a partner and chief scientist with Advanced Solutions Life Sciences, with more than 25 years of experience in basic and applied biological sciences with a focus in tissue biology and tissue vascularization. Previously, he was professor and chief of the Division of Cardiovascular Therapeutics at the Cardiovascular Innovation Institute, where he developed a broad background in tissue fabrication, cell therapeutics, and translation of discoveries to industry and the clinic. Dr. Hoying is an inventor of the angiomics microvessel technology and holds numerous patents related to vascularizing tissues and related cell therapies. He is one of the first to publish in the area of vascularized tissue model fabrication using bioprinting. Dr. Hoying received his BA and MS degrees in biology and molecular biology from Case Western University, Reserve University, and his Ph.D. in cardiovascular physiology with an emphasis on microcirculation from the University of Arizona. Today, Jay will present effective biomanufacturing solutions, which will ultimately depend on automated flexible fabrication approaches. Advanced Solutions uses bioassembly biofabrication platform to develop robotic workflows to fabricate a wide variety of vascularized tissues and tissue models, including thick, high cell density systems. Dr. Jay Hoying will also discuss strategies that involve the integration of 3D printing and other fabrication methods with robotic controls to design, fabricate, and analyze tissue models, such as liver, bone, and pancreas, in a semi-automated fashion. Welcome, Jay. You may begin your presentation.
Thank you, Billy D, for the introduction, and also thank you and Ben Wong for the invitation to be a part of the series. It's a pleasure to be here and given the chance here to talk about some of the technologies and the solutions that we're bringing forward within Advanced Solutions Life Sciences. Back in 2013 is when life sciences first formally started, and it was really in a response in an effort to realize the impact that regenerative medicine could have on healthcare. Primarily to set the stage, Most therapeutics right now are addressing symptomatic type dynamics, particularly with degenerative and chronic diseases. Really, we're managing the patient's health state as opposed to directly curing or fixing the problem. And that's really been, I think, the promise, certainly what the effort that a lot of groups are working towards to realize is that perhaps we can use regenerative medicine to bring about curative treatments, such as one, we can fix the problem. Two, that can reduce costs substantially, certainly for those chronic long-term diseases where costs continue to go on for years, and then improve quality of life as well. And this was really the context that us in life sciences were started thinking about how best to realize that, how best to support the regenerative medicine. We know that it's a growing effort based on a number of different studies and reports. This is a particular one that's available on the web, where they're looking at, in this case, global markets, and by 2026, a $1.6 billion effort. It's collected. Regenerative medicine means perhaps many things to many people, but generally it's built around cell therapeutics, including gene therapies, using some blood products and primary patient-derived products, and then also building tissues, collectively known as tissue engineering. And it's really in all of these that we think we have something to offer, something to enable others to advance the technologies and the solutions. But today, primarily focusing on building tissues, either as a tissue model for drug discovery, for research, basic science efforts, and therapeutics. So, in thinking about how best to move forward and where we can best contribute, we start talking about building tissues, whether for tissue models or for therapeutics. It really starts from this paradigm, such that we start with some cell source. Of course, organs are made from tissues. Tissues are made from cells. So cells are the business end of our efforts here. Really, I think the promise of regenerative medicine emerged when the stem cell technologies started to come out as well, with the idea that we can start to derive from a given patient, if needed, a variety of different cell types. With those cell types, we can grow enough of them to start to build tissues and build organs. And it really is looking at kind of this core stem cell approach. Now, we may also combine that with other types of cells from donors, primary cells. Regardless, it's assembling all these different cell types, either from derived or collected, into those final tissue forms. And beyond the underlying biology to do the derivations and the tissue biology, one of the key challenges to fully realize the potential here is, how do we start to fabricate and manufacture within this context, within this paradigm? And that's an area that we thought that we could contribute and really bring forward some enabling solutions. Also, in the context of that approach, is there have been labs, for years now, across the country, across the globe, developing tissues for a variety of different applications by hand. And so walk back in the lab, back in the tissue culture space, someone's under the hood, taking cells, expanding them, collecting them, packaging them, putting them in devices, combining them with other things. So we have a lot of breakthroughs that are going on with respect to how and what tissues to build. But these are still kind of artisanal. They're made one at a time. They're often specialized approaches to do that. They're very person intensive. And to us, this was really the dynamic that we stepped into and took a look at in considering, how best to move forward. So the challenge then becomes, how do we take these laboratory breakthroughs, both current ones and then those that are on the horizon that are emerging, and really get them into manufactured tissue products. In other words, built with some consistency, with robustness, and to scale in order to really have an impact on the healthcare dynamics that really regenerative medicine has the potential to impact significantly. So for tissue manufacturing, then, some concepts that we were starting to consider and wanting to explore, really setting the stage for how we move forward with this idea that most tissues are built by hand. How do we scale this? And there are a couple of unique dynamics that we think are specific or relevant to manufacturing tissues. And one is this concept of scale out versus scale up. And here, what we're dealing with are biological systems. We're dealing with person-to-person variability. The concept here, for example, what we'd really need is a facility or a platform or a technology base that would let us make 1,000 bone grafts, recognizing that each one of those bone grafts may be distinct and unique. And so there's very much a customization approach to this. It may be the same general type of bone graft, say a segmental defect in a craniofacial application, but because of the defect, shape, and geometry, because of the anatomy of the patient, because of the patient's own genetics and biology, each one's going to be a little bit different. So we definitely have to have that in mind. Very analogous to some of the custom agile manufacturing that is being explored to make bespoke products in the non-living, non-biological arenas. The other side of that, which is somewhat parallel to that and relevant, is that the biological systems are variable and very dynamic. In source materials, certainly with primary cells, we deal with working with primary hepatocytes, liver cells. Those are coming from donors. No two lots are identical. They're still liver cells. They do what liver cells do, but how you use them and how you grow them can vary. Tissue components, etc. The manufacturing dynamics are also somewhat of a challenge in that some cells are going to grow faster than others. Some tissues will form a little bit differently than others. And really, this is what we consider to be analogous to the adaptive manufacturing, which is really manufacturing to scale with robustness, but recognizing we may have to make changes on the fly to realize that final product and outcome. Furthermore, what we wanted to do in our solution in bringing forward a biomanufacturing platform is, building one tissue type, say the kidney, more specifically the nephron versus bone or even heart tissue, may not use the same fabrication approach. So we want our system, our platform, to be flexible and accommodate a variety of different ways, whether it's bioprinting, whether it's fiber pulling, whether it's sheet forming, whether it's any type of specialized approach. Again, trying to honor those artisanal strategies that laboratories are using to build their particular tissue of interest. We want it agile. We want to be able to customize. We want to be able to handle a lot variability that comes with, one, the biological sources, but also different sources for that. We want it adaptive eventually. We recognize that the biology wants to change. It's going to change, and so we want to accommodate that. And of course, we want to scale it, and the best way to do that is through automation. And finally, when we sat back and thought about who the customer, the target customer might be in finally producing something that will enable a tissue to be built, we really focused on the surgeon and the clinician, as these are the individuals that are dealing with the patient, that we're using the tissue product as it's made, and really that frontline, that user case. So we spent a lot of time talking with the clinicians and the surgeons, the so-called end users, to really understand what their needs are. So within that context, our solution really is hey, we want to automate, we want flexibility, we want agility, and we want to leverage some of the pre-existing automation and manufacturing capabilities that are already out there for non-living systems. And so the strategy really came out from all those discussions was, let's use a robotic-based fabrication and manufacturing platform to build these tissues. And robotics and automation are proven. They're constantly evolving, and we think that there's a good opportunity here, configuring this in a proper way, that we really take advantage of all that. And so, to highlight that, back in the early 50s or mid 50s, the first industrial robot was invented by a guy by the name of George Duval. Very large, very heavy, a handful of very discrete execution tasks, pick and place, maybe some spot welding. Since then, we've seen innovations over the 60 plus years where we have some very sophisticated robotic arm-based automation. On the right, you can see this very, active workstation where robots are doing a number of different tasks, a number of different types of robots. And this was really the strength and the power that we wanted to leverage and bring forward in building the tissues. So with that in mind, we brought forward what we call the Bioassembly Biomanufacturing platform. It's really a single platform, flexible enough to accommodate a variety of fabrication approaches. It consists of fundamentally, and as I go into this, you'll see that there's more that hangs off this core technology, is our design software, which is an intuitive platform, takes patient data and lets me design the tissue type that I want and bring in multiple components and manage how those components are used. In our robotics platform, this is a 6-axis robotic arm. Your shoulder and arm, for example, is 7-axis, so we have a lot of freedom of movement. We can swap out the hand with different tools to create different functionalities and really start to do a series of tasks assembled into a single manufacturing run or workflow to start to build those tissues. To coordinate all of that is our design and make strategy, where our TSIM software, coupled with this robot, the bioassembly bot, we design and make using another software platform called the BioApps, where we control all those different tasks. So envision the person, the graduate student or the postdoc underneath the hood, building a tissue by hand. There are a number of different tasks, a number of different steps. These are the elements of the workflow that we need to automate. And our BioApps really coordinates and establishes when those tasks are done, how they're done, and in coordination with other events that are going on. So such that the idea here then is we have one platform, a design, a fabricator, and a make control software that will really let us to do a variety of different applications, whether it's tissue models, vascularization strategies, a variety of different therapeutics, bone, and eventually organs. Real quickly, our software, it's 3D CAD for life scientists. It was very important that the software would be usable by a variety of different expertises. In other words, you don't need to be a CAD engineer or a design engineer to run it. Back in my lab, when we were first working with our first prototype printer, we were scripting everything. We had an engineer running that. He graduated and moved on and left us poor life scientists struggling with how to write this up. So the software here then is point and click. It's intuitive. Lets me take medical images in and design relative to the anatomy and the geometry of the particular defect. In this case, it's the upper jaw of a patient. And then I can build sub-assemblies. I can have multiple parts that are all part of this. Whether it's a small tissue model that has two components, two cell components, or something more sophisticated like a vascularized implantable tissue graft. This works in coordination with the bioassembly bot. Again, as I mentioned, flexible. We want precise control. Again, the robot affords this. We want to be able to interact with other technologies. We recognize that there are multiple different needs, technologically speaking, whether it's analytics, as I mentioned, different fabrication approaches, sourcing, cell manufacturing, cell expanders, and then, of course, the incubation and bioreactor systems that the tissues need to be grown in. And then finally, we can automate it. We can talk with it. This is a robot that is very much learns as we bake in AI components to this. And the user gets to instruct and teach the robot to do the various tasks specific to their tissue type. So it's a platform that can 1 week build one tissue, break it down, the next week build a second tissue, break it down, the next week build a third tissue, and so on. So we really envision, we think that this is meeting our vision of having that broadly applicable, flexible platform. But we've been fortunate enough to be recognized for the technology and the innovation. In 2017, the YPO Global Innovation Award, which was really a competition amongst many technology groups around the world. And then recently, just last year, Frost & Sullivan identified us as the best practices. In other words, they really appreciated how we're trying to bring our technology platform forward for others to use. The BioApp software, as I remember, this is our control platform. This is what's instructing the bioassembly bot to execute those variety of tasks. So here we're instructing, in this case, a 384 well in the context of drug discovery and tissue modeling. Those are the wells I want my construct to be built in. Once that is done, I pull the plate over. I start to build that construct. In this case, it's a 3D bioprinted element. And now I need to have that scanned and looked at. We're working with Cytiva, formerly GE Healthcare Life Sciences, and their high content analysis scanner to automatically integrate, which I'll show you more of that in a little bit. Here, BioApps is asking that imager now to run a scan and give me results so that the workflow decision paths can be executed. And what do I do now? Did it pass what I wanted? It did. I'm going to send it over to our incubator system, the biostorage bot, and then continue with the fabrication approach. This, by the way, has been funded through the Army grant, which I'll talk about a little bit, but certainly we have some federal dollars to help bring this forward. So before I go on, I'll pause a little bit here for any questions. As before I start talking about how we're starting to take this technology platform and start to build those automation workflows. Okay, so for the next few slides, some examples of how we're using the platform, and primarily I'm showing you what's being done in-house here in our R&D facilities, but certainly we're working closely with others around the world to develop their particular specific applications, all with the idea of elevating this solution to enable building tissues and tissue models. As I mentioned, we had partnered with Cytiva in their in-cell analyzer, which is a high content confocal based scanner. Our vision here was we need to have some type of analytics. In this case, our focus is on confocal imaging. But you can imagine having this with ultrasound, perhaps some spectroscopy that might be used. But this was our first use case to demonstrate this automation workflow and integrating different technology types. Here is our bioassembly bot. It's coupled to the in-cell scanner. Here is a black system, and we have this nice interface where the robot can reach out of its fabrication cabinet, drop whatever needs to be scanned into the scanner, and then have our software execute a demand. This is an example of such workflow. In this particular application, the customer is wanting to have test a variety of different cells in a 3D alginate gelatin matrix environment. Here the plate is brought into place. This particular lot of cells in the matrix is deposited, not printed in this case, but it could be printed. Putting that plate back together, and we're going to incubate this for a little bit. And this is prescribed, and this is where perhaps we incubate for 2 days, take it out, bring it to the scanner, take a look. Are cells progressing? Are they dying? We can take measurements. With our AI platform, which I'll talk about later, those measurements can be made on the fly and logged such that you have a running collection of data. Here we're aspirating. We're going to be putting new media on at different steps. We're going to be adding dyes to do tracing. So hopefully you can start to appreciate those that have been in the lab working under the hood. We're really starting to replicate those tasks that you might do to execute this assay. Here we're adding media to it. So this might be part of a feed. And then we're going to add a dye here so that we can start to count nuclei. And all this can be incubated. Pick up the plate, transfer it to the scanner, instruct the scanner to acquire its image. Here we have nuclear. Each little blue dot is a nucleus, and then we continue the assay, whatever that might be. And that data now has been downloaded and being processed for further analysis. And maybe I need to grow the cells a little bit longer. Maybe I haven't reached the critical cell density yet for my particular application, so I might want to incubate it further. So this is designed to give you some concept of what we mean by a workflow, and then a combined integrated work cell where all the different technologies needed to execute that task are being done. And as I mentioned, there are a number of applications this is being explored in, whether it's medical devices and using biocompatible materials to build structures and devices, therapeutics, tissue models, as I mentioned. We're starting to get heavily into spheroid and organoid processing and management. I'll show you an example of that later as well. It's exciting because our initial vision back in 2013 was to have this nice flexible platform that can be applied everywhere. And here in 2020, we're starting to realize that and see it being used in a variety of different ways, even ways we didn't really imagine, 7 years ago. So what I'd like to do next is take you through some examples where we're using the platform in combination with our angiomics vascularization solution to start to build vascularized tissues. So right now, some of these are semi-automated, but our next stage is to, once we develop the protocols, to translate to a fully automated solution. And so as we go through this, I'll try to point out where those areas of automation or potential automation will occur. Step back a little bit and just set the stage here. I've been talking about really our hardware software components to what we call our technology stack. Really, as I mentioned, our original vision was to establish this platform to enable then the fabrication of tissues, again, for a variety of applications, but also starting to bring forward our vascularization solution. We think we have a unique solution, an effective one, for vascularizing tissues. I'll show you an example of that here in a minute. And really, our workflow is from the ground up here to finally realize, these final tissue products. Unlike more traditional approaches where we try to build vessel segments from scratch from endothelial cells or other cells that make up the normal vessel structure, we decided to go with what Mother Nature uses, which is the intact vasculature or intact individual vessel elements. Our source here is adipose. We strip away the adipocytes, in this case the red balls of ball-like cells. We have our green vasculature here. During processing, that gets fragmented into individual broken vessel segments. These are whole intact microvessels, arterioles, capillaries, and venules, for those in the know. They have all the requisite cells necessary to build a mature vessel segment. This case here, we have a post-arterial precapillary segment of the microvasculature. And hopefully you can appreciate, we see a variety of different cell markers here. We were somewhat surprised to realize that all of these different cell types are associated and help make up the wall of the vessel segment. And so we think that this vascular complexity is very important and really enables us to start to recapitulate that native vascularization process that happens in the tissues. And so really our strategy is to recapitulate as much of that happens in the body in the laboratory. For example, here, show you a time-lapse video of new vessels growing from these vessel segments that we've cast into a 3D tissue environment. This is over 6 days. The red here represent the vessels that we've harvested. The green is the fibrillar matrix of the collagen, in this case, that the vessels are in. And here's one of those vessel segments with a new vessel segment, a neo vessel growing out from that. You'll see that it'll start one direction, back up, change direction, and then find and connect a partner that's growing into the field of view here to make this one contiguous segment. This is advantageous in that the system in and of itself, through a bona fide sprouting angiogenic event, will create a network of vessels comprised of all those cells needed to eventually mature into a final microcirculation once it's given the right hemodynamic cues. For example, we can take this isolate here of a variety of different vessel segments, we can expand them in culture to form this interconnected neovasculature, and when we implant these, in this case, these were implanted in the sub-Q position of mice, we can derive a variety of different microcirculation phenotypes and topologies. This is one type unit that was derived from the system, and here 2 microvascular units came out of that single entity. What this reflects, though, is really the ability of that neovasculature to read the tissue environment and to build a microcirculation that matches the tissue needs, whether it's in a liver context, muscle, perhaps a blood-brain barrier-like phenotype. The cell complexity that comes in with the vessels enables this kind of adaptation, and in the end, lets the biology do all the heavy lifting, and what we really need to do is control that environment. So with that in mind, we wanted to leverage that intrinsic dynamic. All we really have to do, easier said than done, of course, is define that tissue environment to let the biology do its thing. And so the first real effort towards this was in the context of building a thick vascularized liver model. This liver tissue is about 1 centimeter thick. We can go bigger and thicker. We've done that, but traditionally we're dealing with about 1 centimeter. And what we really focus on is organizing the environment that enables the hepatocytes, the non-parenchymal cells of the liver, as well as our vessels shown here in purple, where all of the other liver cell types are in green, to coordinate and assemble, keep them alive and functional. Interestingly, what we've found is with our microvessels, even though there's no blood flow to the system yet, the tissue behaves better. It stabilizes sooner, as shown here by LDH release activity. Here, no vessels, here's with vessels, and we have much less cell loss early on. Both systems eventually normalize out, but we can get to a healthy liver tissue sooner with our vessels. And in the end, we're starting to see more functional phenotype by our prevascularized livers with just having those vessels there. And we think, again, because all those different cell types are really contributing to that more complete tissue environment. Because we're dealing with the tissue environment here, we have opportunities to reconfigure this in a variety of ways, such as using our bioprinting system to incorporate a sacrificial perfusion channel network that we print up within the TSIM environment. The bioassembly bot will execute the workflow, print this up, combine our cells and matrix, cast the tissue around it. Once that tissue sets, we wash out, leaving behind our flow channels formed by the tissue itself, and we can cellularize this. So we can start to really configure this liver tissue in a variety of different formats depending on application and need. As I mentioned, we're starting to work with spheroids and organoids for a variety of different applications. And our focus has been how to vascularize those, how to build a vascular system within that spheroid, again, based on some of what we're learning in the liver, where the vessels contribute to a more improved tissue environment. In this particular experiment, we found that there is an optimal ratio of spheroid cells, in this case, these are mesenchymal stem cells, to microvessels, in this case, a 50-to-1. And we get nice vascularization. If you take that pre-vascularized spheroid, place it in a 3D tissue environment, and let it undergo additional culturing, we get nice vessel outgrowth as the system starts to undergo angiogenesis, again, looking for a vasculature to connect to. And so we think this is a universal ratio that will work for a variety of different parenchymal cells, and we're exploring that now with other partners. Hopefully we'll have some of this work out published in the public in the next couple of months. In our efforts towards therapeutics, we've been developing in-house a customizable approach to making bespoke bone grafts, what we call a biobone solution. This here is responding really to the need, such as you might see in craniofacial applications, where the geometry of the defect, and here is a model, lower jaw, a mandible, where the geometry of the particular defect is going to be very irregular, very unique to that patient. And what we want to be able to do is to build a bone graft. In this case, there are no synthetic elements. It all has biological materials with it that we can shape and then pre-vascularize to enable engraftment. And so we have a solution where we can print up a core or wrap it with a vascularized tissue shell using our angiomics technology, and really create something that can be used by the surgeon to just drop in that patient's defect, which, by the way, we're using scans from that patient beforehand to do the design and establish that initial geometry. So this has really been an exciting project, and I think is opening up a lot of opportunities for us from a manufacturing perspective. Finally, the one project that we've been putting a lot of effort in is, as I mentioned before, we can pre-grow our microvessels to form a neovascular network. When we implant that into an animal, say sub-Q in a mouse, the neovasculature we built will connect to the host circulation. Blood will start to move through that neovasculature, and it will eventually drive to a mature microcirculation. We want to capture that dynamic in vitro and build a human microcirculation. So our strategy has been to combine the microfluidic channels, similar to what we showed you in with the liver construct, get our channels here, which are inflow, ways to get fluid in and out of the tissue environment, and then have that coupled to our neovasculature, eventually driving maturation of that microcirculation. We're pretty far along on this project. We certainly know how to build channels that are cellularized. We know how to grow our microvessels. We're getting a nice network formation with that neovasculature. Down here is a lower mag showing here's our channel boundary. This is part of the device that we use. I was hoping you could appreciate we have this nice neovascular network surrounding that channel. We know that they're connecting to that channel. Here's an example of that inosculation event. And just a couple of weeks ago, we had our first real bit of preliminary evidence that we're getting perfusion through that neovasculature. So we think we're well on our way to start to drive this maturation and form this microcirculation. To us, that opens up a variety of different possibilities. Even from a tissue modeling perspective, imagine dropping this pre-vascularized angiogenic spheroid, whether it's built with MSCs or perhaps tumor cells or perhaps stem cells deriving a variety of different cell types of interest, into the context of this network, having it integrate with this neovasculature. This neovasculature integrates with the channel, and now we can start to really build tissues that are perfused in vitro, analogous to how they happen in the native environment. And so we're really excited about this. Hopefully in the next couple of months, we'll have this, the data collected to get this published and out for people to start to think about and use. So I'll pause here for a little bit if there are any questions before I change gears a little bit and get into the context of adaptive manufacturing. what I've shown you are those variety of applications that we're using in-house to help develop our technology stack, our hardware and software solutions to automate the fabrication and manufacturing of tissues. Also, we're learning a lot about these tissue models and the tissue biology that's important in building tissues. And so now really it's to the point where we're starting to get in and advance really this concept of manufacturing for eventual clinical use and application. So to talk about adaptive manufacturing here, and really I think this is also, I won't say necessarily, but certainly I think involves in a significant way artificial intelligence and some kind of smart element to that manufacturing process, either in the instruction, assessment, or the decision making. And so the adaptive manufacturing, at least in our minds, it's a system that will adapt in real time as it's building the product of interest. It invariably involves computer vision. The system needs to see what it's making, to see how that is going, to accommodate any differences you might have. As I mentioned, biological systems are variable. They don't grow all grow the same. They don't necessarily start all the same. And so we want some way to accommodate that. And as I mentioned, that process is not constant. So if we want to fully automate, we need to have something in place that will monitor, assess, and make a decision based on how the tissue is forming. And a real life example of adaptive manufacturing is coming out of, I've learned, finishing blades for jet engines. So these are those vanes and propellers that sit within the jet engine. And apparently that's typically done by hand because there's tremendous part variability and there's very tight tolerances. And there's efforts now to automate that by hand part using AI to assess, grind down edges of the propeller or the vane for the jet engine, monitor, we need to do a bit more, monitor, need to do a bit more, et cetera. To me, it's very analogous to what we have to do with building tissues and manufacturing to scale of those tissues. And then implied in that then is some way to assess quality assurance such that we're getting the product that we want and need consistently. And again, the concept here is the biological systems are inherently variable and changing. It's an intrinsic feature of biology. I think what makes the biology so powerful, but at the same time challenging to automate in the manufacturing. And as I mentioned, we're baking AI really into a variety of different components of that workflow. One area is in fabrication error detection. So again, using bioprinting as a example fabrication approach. This is real-time assessment of some of bioprinting applications that we've mocked up and recreating common errors such as bubbles, layers aren't adhering well, sags and defects that might happen. In this case, bulging where your needle is sticking too far down in the material, etc. And here, this is coming out of our AI division within the company, a very effective group, knows AI well and how to apply, and really starting to do real-time assessment during the fabrication run, such that, are these errors within specs? Are they acceptable? Or is this a failure? All of that is part of the process that normally a person does in real time back in their laboratory. At the same time, we want to do assessments of the biology as it's growing and forming and maturing. Again, we have an emphasis on vascularizing tissue. So our first effort here is in assessing the vasculature as it grows. This is a growing neovasculature, and we're doing automatic vessel detection, and then coupling that with AI analytics to do feature assessment as well as vessel length measurements, for example, one of a common measure analytic that we rely on. We've done some comparisons off of our first round of our AI system, which we call VSA. I'm very excited about it. I think it's really proving extremely useful for us in-house. And we compared it really to this kind of standard process here of take an image, you annotate, and can we get the AI to match the annotation done by hand. So in this case, this middle column, someone has gone in and traced each individual vessel segment. You might imagine it's laborious. And it's a person, meaning Monday may give a different annotation result than Friday or Wednesday. And we really looked at two different applications, fluorescence here via confocal, and then phase, which is notoriously difficult to automate in assessment and measurement. So having said that, we compared data between the person annotated, which is a black bar, that which is done with the AI, which is the gray bar, and then the traditional intensity-based thresholding pixel-based approach, which is the white bar. We see pretty good matching between the VSA, but the pixel-based did, just as well in low mag confocal. But as we go to higher magnification, in this case, this was done specifically with our in-cell scanner partners equipment, Cytiva's nice system. We're seeing very good association between the annotated here and the VSA, the AI, and the pixel-based system just really struggles. Interestingly, in this particular data set, the person doing the annotation took them about 5 hours to do 45 images. The AI system did it in under a minute and a half. So the time benefit is tremendous as well. And we even had good success using the AI system to assess phase, which is notoriously difficult to do. The pixel-based system simply failed. We weren't able to get any legitimate data out of that. And so our accuracies are very acceptable. They're certainly as accurate as what we think the person is. And so this right here now is being baked into our bio apps, our automated task execution workflow software as part of that automation effort. And some of the examples I showed, we were scanning plates. We'll have this AI system sitting in the background, reading, monitoring, measuring, and then sending information for decision making in the automation. And that's really what I'm trying to highlight here is that we integrate these different analytics. We use AI then to, in the context of automated workflow, to make a variety of different measurements and leveraging, in some cases, Cytiva's very powerful image analysis software to do even more advanced analytics than what we've been doing. So with that in mind, my last few slides here are really based on that context that we've just talked about, maybe set a stage for what we think the near future is going to involve and how we hope to see our automation platform be used by others and integrated into some of these broader applications. And two examples I'll show you here is an in situ biofabrication, and then our concept here of a clinical biofactory. So working with partners at the University of Lyon in France, Dr. Christophe Marquette, a very innovative, very forward-thinking group, using our platform to do in situ, what we call in situ tissue fabrication. In this case, they work in skin, primarily in pediatrics burn arena, where they're trying to help kids that have suffered significant burns, and to, in this particular application, come up with a regenerative medicine solution for skin that can be applied directly to the patient as needed. There are a lot of implications here. And it's a very exciting, emerging, still a lot of things to map out. But nonetheless, we work closely with Christophe's team to modify our bioassembly bot. Hopefully you can appreciate, here's that robotic arm. In this case, instead of reaching out the side to the in-cell scanner, it's reaching out the back to, in this case, an animal, a pig model. And this indeed has been done. Here's an image where the T-SIM is showing that patch to be printed. We scan the surface of the pig to provide the digital context to design the patch within. Mocked up different examples to get our parameters and settings, and here it is still from the process where it's actually printing into the wound. Christophe's team is getting ready to get this published. It's under peer review right now. Very exciting application, and I think starts to show what some of the capabilities are here when we start thinking in terms of automating a lot of this regenerative medicine process. In our particular case, again, coming up with an automation to build these tissues, We're really working with a number of different pieces, parts for this. We have patient imaging that's driving our design. As I mentioned, our platform here is working with a number of different ancillary systems, whether it's our biostorage bot incubation platform, which I haven't talked that much about, whether it's a self-sourcing solution, ways to bring in consumables and medias. Perhaps we need to bring in different parts of a tissue and assemble that into the bigger organ. And then, of course, assessment. And really, we've been working closely with understanding how to integrate these elements into this assembly platform. I've given you examples of how we're integrating different analytics into this platform. Ultimately, we have a bio app in the case of our liver that does this integration, executes this task, such that in the end, we end up with our desired liver tissue. And so, I've shown you parts of this overall workflow, and we're very close to, I think, showing a first prototype application like this. Ultimately, though, we envision this broader clinical biofactory where our bioassembly platform is part of this larger engine where patients are scanned. If there are cell collections that need to happen, there's modeling that happens, both the tissue design, perhaps also model the workflow dynamic, as well as modeling the process. Really where a lot of that, the pre components, the cell expansion, vessel harvest and growth, and then finally back to the assembly bot, which then gets passed on to the surgeon to implement. And so we really envision, this broader application somewhere down the road. Built into this is a tremendous amount of opportunity and innovation in manufacturing. How do you source? How do you manage the sourcing? How do you quality control? How do you oversee the manufacturing? Who's doing the manufacturing? All of these, I think, are very exciting opportunities as something like this starts to be realized. So with that in mind, I thank you for your time. And leave it with the idea here. It's one of my favorite cartoons where there's a patient talking with a doctor, and he's saying, "Well, doctor and physician are outdated terms. I'm your biological tech support specialist." And so I think that sets the context really for this broader kind of integrated science, engineering, manufacturing, and medicine dynamic, where really it becomes something more than what we're seeing now. And hopefully we realize that regenerative medicine promise that was laid out some years ago in the near future. So again, thank you, and I'm happy to entertain questions.
Thank you very much, Dr. Hoying, for that fantastic talk. Very exciting work that you're doing. Actually, right now, I don't see any questions in our Q&A panel. I'm going to ask that anyone who has questions, please submit those as soon as possible, and we will get those answered. While that's happening, I just want to remind everyone and actually let you all know that we are recording the lectures and that they will be made available. We're actually uploading them to YouTube with the speaker's permission. And we actually will post them on our website at manufacturing.godtech.edu/lectures. So there will be a link posted to our website to the YouTube video.
And Billy, do you know how to get a hold of me should others have questions afterwards?
Yes, sir. Yes, sir. All right. So if there are no further questions, then Jay, we really thank you again for the exciting talk, and it's time to wrap up. So I thank everyone for attending this lecture series. We look forward to having you again next week. Next week, we'll actually have Kelly Ballou from Shaw Industries going to be speaking about sustainability. And we thank you very much. I do see one question that just popped up. Okay, a link in the chat. Sure, I can do that. So I've posted a link in the chat. to take you to the recording. It probably will take it may take 2 or 3 days before the recording is uploaded. So give it some time.
So Billy D, I see one question.
Yeah, we just had a couple of questions come in. Dr. Shrees Melko says, very nice talk and exciting work. From a standpoint of automation, hardware, and software, what additional innovations from an R&D standpoint do we need going forward?
Yeah, I think it's a spot on question, and I think where we're seeing others start to innovate is in different ways to actually a number of different places. One is in again going back to the idea that someone under the lab has come up with a special way to fabricate the particular part of their tissue of interest. That works great there under the hood, but how do you translate that often maybe multi-step fabrication process into something that can be automated? And so that might re-envision how you are doing sheet extrusion, or how you might spin fibers that you're going to bundle together. So I think there's a real opportunity from a hardware software in that regard. Of course, there's so much room in the AI and workflow management, and also platforms that can talk to each other and communicate. I think those are important areas. And one area which isn't necessarily specific to what you're talking about, but I think it's relevant, is in quality release criteria. So quality assurance, quality control. If you think in the terms of adaptive manufacturing, that is every time I build, for example, the bone graft, it doesn't quite go the way it did before. How do you assess that? What are the criteria? How do you make those measurements? So, the analytics you might use. We're exploring using confocal scanner. We're starting to explore the use of ultrasound, but there's a whole spectrum of ways to assess that could be used, I think, to set these new kinds of dynamic quality control features and parameters.
All right. Thank you for that question. We have, Jay, were you going to say something else?
Well, I was just going to say, and then from the hardware software side, but of course there's all kinds of room in the biology side, whether it's cell sourcing, whether it's materials, et cetera. So it's really kind of a multi-part opportunity here.
Excellent, excellent. We have another question. Any control of material properties like electrical properties?
That's right. I think there's a lot of innovation that's happening there. For example, we had a conversation with a customer that was asking about 3D bioprinting a conductive material or matrix that they could use to paste a system. I think they were looking at a research application, but certainly it's not a far stretch to start thinking about therapeutics. Whether it's conductive, whether it's perhaps reporting. The FDA is starting to look about having built-in reporter systems as a way to monitor. I think there's a lot of opportunity to start to functionalize, from a tissue function perspective, materials that are used to build the tissue.
All right, we have another question. Dr. Kevin Wong says, great presentation. Could you give more details on how advanced solutions address the challenge of printing scaffolds across different size scales in the same tissue?
Yeah, that's right. So I kind of glossed over that, focusing more on the manufacturing, but some of our very early efforts were about accommodating different printing applications. And so that includes different materials, different scaffold configurations, both at a very small scale and a very large scale. So one of the advantages of our robotic system is that we have a very large work envelope. So we have customers that are making structures that are, 10 centimeter cube, for example. Everything from, a bone-like matrix to PCL to silicone to other types of applications. And our software lets you dial in infill patterns, for example, on the fly. It lets you dial in different parameters and controls. And then finally, that robotic arm lets you swap the hand. So I can use a very high resolution, print head, or I can use a very low resolution, high volume print head, a heated tool, a cool tool, a dual mixing tool. We have nearly 10 or so different types of print heads to accommodate this very question, to give me flexibility in how I want to put those scaffolds together. And you can see examples of all that at our website. I think we have videos up, and I think it does a good job presenting some of those opportunities.
All right. Thanks, everybody. It's past 1 o'clock now, so I think we're going to wrap up the session. Again, I mentioned, please come back next. We have Kelly Ballou from Shaw Industries will present on sustainability. And we thank our speaker today one last time. I thought it was an excellent presentation.
Thank you, Billy. It was a pleasure to be here. Really enjoyed it.
Awesome. Awesome. All right. We're going to close. Happy Monday, everybody.
You too. Bye.
Bye-bye.